Good morning, everyone. I am Ashish, Vice President with Zydus, and at the outset, I thank Mumbai Hematology Group for giving Zydus this opportunity to collaborate for today's insightful webinar on use of real-world evidence in research. And I welcome the eminent guest speaker for today, Professor Tarek Christopher from Denmark. A very warm welcome to our chief guest, Professor Shripad Banavali from TMH Mumbai. We also have a very august group of Indian faculties as discussants for this very important topic. And I also welcome virtual delegates who have joined us not from this India, but from various parts of the world. Now, a very special welcome to the person who has brought everyone of us together for today's webinar, Professor Dr. M.B. Agarwal. He has been at the forefront in driving scientific initiatives over years with only goal of improving hematology care in India. While he's spearheading this scientific symposium with never-ending energy, his life has also inspired so many young clinicians to take up hematology practice. As we all know him, not only he is clinician par excellence, but teacher and guide for so many clinicians in India. I have the privilege today to introduce Professor Dr. M.B. Agarwal. Sir is Professor and Head Department of Hematology, Bombay Hospital Institute of Medical Sciences, Mumbai, and also consultant at Leelavati Hospital and Breach Candy Hospital. With that, sir, I would request you to take the program forward. Over to you, sir.
Thank you so much, Mr. Ashish, for those kind words. I'll just share my slide deck. So good morning to one and all. Today our guest speaker is Professor Tarek Christopher L. Galili from Denmark. He will be lecturing us on use of real-world evidence in research. An interesting and unusual topic for these webinars, but I've gone through his slide deck and it's very, very exciting. And I'm really looking forward to what he's going to present and what he's going to discuss with the researchers in the field of hematology from India. This webinar is brought to you by Mumbai Hematology Group. It is supported by Zydus and it is managed by Mice Ideas. I thank Mr. Ashish Kumar and his team from Zydus Kedila for supporting not only this webinar, but number of our academic activities throughout the year. Rajesh, Kalpesh and their team from Mice Ideas for beautifully managing flawlessly all these episodes of live webinars of MHG. We've done almost 150 of them in last 1.5 years. The Executive Committee of Mumbai Hematology Group for giving me this totally free hand to organize whatever academic activities that I wish to. Our guest speaker, Professor Tarek Christopher L. Galili from Denmark. All our discussants who are themselves eminent researchers in the field of hematology and medical oncology from India, and you participants from all over the world for sparing your Sunday morning. Once again, we have almost 500 plus registration for this event. Majority from India, but of course, almost 150 from outside the country, not only Asia, but from the rest of the world. And there are more than 100 fellows and trainees who have logged in. Their future will be much better after listening to this talk. Just to introduce you to the next weekend activity, Saturday, we have Dr. Amar Jaden from Yale University, Connecticut, US, speaking to us on novel therapies for myelodysplastic syndrome. So after a long time, we are going to discuss this subject. This is on Saturday, 13th November, 7 p.m. And next day morning, we have from France, Dr. Thierry Lemy, who is going to speak to us on LGL leukemia. And once again, for the first time, we are going to discuss LGL leukemia in this series of webinars. And this will be on Sunday, 14th November at 11:30 a.m. To introduce our guest speaker, Professor Tarek Christopher L. Galili this morning. He is currently professor at Aalborg University Hospital, Denmark. He is a consultant hematologist working primarily in the field of lymphoma. His primary research focuses on clinical epidemiology and generating real-world evidence within the field of hematology as well as PET imaging. He leads the epidemiology real-world evidence research group for hematology at the hospital. He's published over 90 papers in peer-reviewed journals, including several papers in high-impact journals. So we are very, very fortunate to have a personality like him speaking to us. And I'm sure one day he will be with us in India talking to us. Let me now introduce our discussants this morning. And we have got our colleagues and researchers from Bangalore, Lucknow, Vellore, Puducherry, Chandigarh, Ludhiana, Kolkata, Ahmedabad, and Thalassery this morning. To introduce them individually, Dr. Govind Babu is from HCG Hospitals, Bangalore. Professor Sonia Nityana from RML Institute of Medical Sciences, Lucknow. Dr. Vikram Matthews from CMC, Willow. Dr. Prashant Ganeshan from JIPMA, Puducherry. Dr. Pankaj Malhotra from PGI, Chandigarh. Dr. Joseph John from CMC, Ludhiana. Dr. Pranta Chakravarti from Fortis Hospital and other hospitals, Kolkata. Dr. Abhishek Kakru from Vedanta Institute, Ahmedabad. Dr. Chandran Nair from Malabar Cancer Center, Thalassery. Before we move further with the guest lecture today, our usual Sunday quiz for the postgraduates. And today this quiz is related to asciminib. We are going to give you 7 statements regarding asciminib. One of the statement is wrong. You have to pick up the wrong statement and email its number to mbagarwal1@gmail.com. Don't have to write the whole statement, just the number 1, 2, 3, etc. Just that number, and whoever is the first to send the right answer will be the winner. So that's fastest finger first. And of course, we are going to give you names of all those whose answers were correct. And this will be immediately after the guest lecture, before we take up the Q&A session. Now, these are the 7 statements regarding the drug asciminib. So first, asciminib is an oral STAMP inhibitor, STAMP standing for specifically targeting the able myristoyl pocket. Number 2, asciminib is indicated as monotherapy for those patients of chronic myeloid leukemia chronic phase who have been previously treated with 2 or more tyrosine kinase inhibitors, or who have developed T315I mutation. Number 3, ASSEMBLE was the registration trial comparing asciminib versus ponatinib in patients of CML chronic phase who have received 2 or more TKIs earlier. Number 4, asciminib is marketed as a film-coated tablet under the name Semblix. Its dose is 40 mg twice a day for CML chronic phase, and the dose is 200 mg twice a day for CML chronic phase having T315I mutation. This drug is best taken on empty stomach. Statement 5, asciminib has no off-target activities and hence it is safe with no major adverse events, especially no significant cardiac toxicity. Number 6, no dose adjustment is needed for those who have mild or even severe hepatic or renal dysfunction. And the last statement is, it is under research in combination with other tyrosine kinase inhibitors like ponatinib or others like imatinib, etc., that act by blocking the ATP binding sites. Such combinations are effective against the compound mutations, and the combinations also permit reduction in the dose of ponatinib, which has a dose-related cardiac toxicity. So these are the 7 statements. I'll just keep it on for 10 seconds. At the top, you have the email ID on which you have to message one of these numbers. And if you're fastest finger first, you will be the winner and others will have their names read out. So please email the number of the false statement to mbagarwal1@gmail.com. Now it's my pleasure to introduce our chief guest for the day. And that is none other than a very popular personality in the field of hematology and pediatric hematology oncology, Dr. Shripad Banawali. You all know him, but I have to complete the formalities. He's Director Academics at the prestigious Tata Memorial Center, Mumbai. He's Professor, Department of Medical and Pediatric Oncology, at Tata Memorial Center and Homi Bhabha National Institute, Mumbai. He did his MD in internal medicine and then underwent adult oncology training at TMH, Mumbai, following which he did a research fellowship in the University of Cincinnati. He did his board certification in pediatrics and fellowship in pediatric hematology oncology at St. Jude. After spending 8 years in US, he came back and joined Tata Memorial Center, Mumbai. He's a member, Subject Expert Committee of the Drugs Controller General of India. He's chairman, Indian Pediatric Oncology Group. He's founder chairman of Impact Foundation. He has developed the cancer facilities at the BKL Valavalkar Hospital, Konkan, the Rural Outreach Program of TMC. His current areas of interest are drug repurposing and development of low-cost, effective metronomic therapies for the treatment of various cancers. He is worldwide known for development of these metronomic therapies in cancer. There's more than 500 scientific publications, papers, and abstracts in various peer-reviewed journals. He's the recipient of various national and international awards. He's traveled widely across the country and the globe, delivering various orations, plenary talks, and lectures. It's my pleasure to request him to inaugurate today's webinar and give us some words of wisdom. Thank you so much. Over to Dr. Sripath. Yeah.
Thank you, sir, and thank you for the honor to be the chief guest of today's seminar. So, as you have rightly told, Dr. Agarwal has really utilized this, the lockdown period, and he has conducted more than 150 seminars and webinars during this period, so that he has utilized nearly every weekend that was available during the last 1.5 to 2 years, and we have covered many really good, subjects during this time. But today's, I think, is a unique seminar there, or webinar that has been conducted, because we usually talk about basically the phase 3 trials, the randomized trials, where we have highly selected patients, and that data is important. But many a times that data gets diluted in the real world scenario, and we have seen that multiple times when we have used drugs for various conditions, and now we understand the importance of the real world data, the real world evidence in research, because that also gives us a lot of information, not only about the disease and its natural course, but also many therapies which may or may not stand the test of time, and many drugs which are found to be effective in the phase 3 trials may lose the importance when we really see the real world data. So especially a lot of students attend these seminars, and this is very important for them to really analyze the papers which are available and the drugs that we use, where even for small basically advantages, we are using a lot of drugs which are really, especially the newer drugs, which are very, very not cost effective, I would say. And when we see this real world data, we come to understand the importance of this type of study. So thank you for keeping this very important topic in a webinar. And we have Professor Tariq Christopher L. Galali, who basically is going to give us the real importance and the insights in how to utilize the real world evidence and data, and maybe after that the discussion that will appear will happen, will throw light on this important topic. So without wasting much of your time, back to you, Dr. Agarwal, and thank you once again for giving me this opportunity, and we'll learn a lot from this webinar. Thank you, sir.
Thank you so much, Dr. Sripath, for those very kind words and for sparing your Sunday morning to be with us and inaugurating this webinar, which, as you said, is extremely important. The subject was selected by Dr. Tariq himself, and we are very excited to listen to him. For those of you who are relatively new in the field of hematology, Denmark is a very important place. I've been to Denmark at a couple of occasions. Last was the visit when IHA was held at Denmark, and you all must visit that place sometime in your life. We have had speakers from Denmark in past, and we were just chatting with Dr. Tariq, with Dr. Martin, who spoke to us on Hodgkin lymphoma, various aspects, and his interest in PET scan also allowed him to speak on PET scan to us. Coming to India, he was our guest at 2 or 3 occasions, and he has conducted webinars for us. And also Dr. Lena Specht, she spoke to us about radiotherapy in various lymphomas, also on the PET scan once again. And this is, I think, the third speaker from Denmark who is going to address us. So now over to Dr. Tarek, please.
So thank you so much, Professor Agarwal, for this kind introduction. I have been really looking forward to this meeting, and I just got the opportunity to hear just before we started how often you have this, and I was almost shocked that you can make this every week. This is simply amazing education program you have. So it's really an inspiration to many other places in the world who really doesn't have this intense education. It must be fantastic for you to have this program every Sunday or Saturday. And I'm really happy to give this presentation. As it was said, I actually chose it myself, but this is of course because this is what I work mostly on and what I feel I have the best hand on among a lot of other subjects. But and it's also getting, as I said, increasingly important to realize that there are differences in our data sources, and clinical trials are not real world data, and real world data are not clinical trials. So I walk you through this. I'll just share my screen, and then I hope we can have a good discussion on this afterwards. So do you see my screen now?
Yes.
And is it in presentation mode?
Yes.
Great. Thank you very much. So the first thing I will go through is talk about real world data and definitions and types of data sources. And then I'll discuss, actually most of the time I will be discussing real world data in drug development, because I think this is a new area, and I think there are some really great potentials, but also some very important pitfalls I will discuss. Then I will also briefly touch upon real world data for pharmacovigilance, and if we have time, then also to inform clinical practice. But this is the last point. We can skip that in the sake of time. So we have a lot of different sources of real world data. We have the typical registries. This is something we have a lot of in Denmark, and as you know, this requires that you register patients over time in many different registries. We have like diagnostic registries or reimbursement and so many other things, and you can actually use this for research even though it wasn't developed for this purpose. Oh, sorry. Then we have the electronic health records, as you also know, or paper records. This is also a typical source of real world data. And these are the two classical forms, but we have so many other things. We have so many patients now having wearables, iPhones that can capture data on physical activity and so many other things, symptoms. We are all part of networks on social media, on LinkedIn, maybe on Facebook, and so on, many other things, which is actually also getting an increasingly important source of real world data. We have the patient-reported outcomes, and we have also, of course, pathology banks from various places where we use the pathology for routine diagnostic purposes, but whatever left over is often left in a pathology bank. So there are so many options to look at real-world data, and so many combinations now. But even though there are so many options and so many combinations, there's actually no standard definition on what is real-world data and what is real-world evidence. This is a paper. It's not. It's from 2017, so of course the definition has moved a little on since then. But this was a paper that looked at the definitions and looked at the literature and asked different experts on what is real-world evidence, how to define it. And what they saw was it could be grouped into 4 categories. There was one category which was data collected outside randomized trials, randomized controlled trials. The second category was data collected outside an interventional control setting. The third category was data collected outside an experimental setting, meaning that there was no de novo data collection. And then there were other categories. So this was the way it was defined. And as you see, there's a huge difference. Like if you have all data sources outside randomized trials being real-world data, then you have a huge variety of data. But on the other hand, if you only have data that is where there was no de novo data collection, then you really have data sets with all the limitations. And and problems of typical real-world data. I'll try to illustrate it here. So if you have like the grading of evidence, if the randomized trial is the highest, for example, and the non-experimental is the lowest setting, this could be, for example, a registry developed for something else where you use it for secondary use. Then you have the single-arm trials, you have the non-interventional trials. And if you have a really wide definition of real-world data, as some have, then you can include the single-arm trial, the uncontrolled setting, the non-interventional trial, and all these things in real-world data. I don't think this is the typical way people look at it. I think more typically people would look at this as being the non-experimental data, the data where we use data collected for some other reasons and do secondary analysis on these. I think FDA has a quite nice definition on it, and it's data regarding the uses of potential benefit or risk of a drug derived from sources other than traditional clinical trials. And this is also how I look at it. Like all the things we have outside the traditional clinical trials, and that be randomized or single arm, this is a type of real world data. I think one important aspect of real world data is that many will have the feeling that real world data is always something that is done retrospectively with all the limitation. And this is actually not the case. Many real world data studies, even those that are analyzed retrospectively, are actually based on prospective data. And this also includes many of the registry studies we do in Denmark, because data is collected continuously all the time. And then at some point you do an analysis. Of course, you do that in a way retrospectively when you have collected all the data, but the data is actually fixed and collected continuously. So real world data is becoming really popular, and you can ask yourself sometimes, is it hype or hope? We have a sentiment today that data is almost like having gold, right? If you have more data, then you're sure to be the winner. But I think there are limitations, because as we know, when we have very big data sets, they are collected yesterday or many years before this, and can we really solve all today's problems by looking at yesterday's data? I'm not sure. We probably need a good combination. And we also have seen a tendency now that we can use real world data maybe to replace the need for randomized trials, because they're getting so expensive and complicated. And what I show you here is also the share prices of a Tesla share, and this is also something that exploded. And I think also for the use of real world data, there's also some inflation in what you can use it for, I would say. Although I have to say I'm not an economist, so I don't know if Tesla is really worth that much. I think what we will need to see in future is that the evidence hierarchy will change for many structural reasons. So we all know this pyramid that we have the randomized controlled trials, or maybe even if feasible, the meta-analysis of randomized controlled trials as the very top of the evidence pyramid. And then we have all the real world data studies below. I still think that the randomized trials are critical and really important, but I think we will move to a scenario where we will have to accept that we will not have these very, very big randomized trials. We may not have 2 randomized trials of the same drug. So we will have to look at the totality of evidence more and more in the future, and not only a single source of evidence. And this is, of course, especially true in hematology, because we are moving too much we're moving towards so refined disease classifications, where we find more and more molecular subgroups, and we also have more and more treatments in development. So there's simply not it's simply not feasible anymore to do the big randomized trials in all diseases, in all settings. I think there are a couple of topics or areas where real world data has a real important application. I think first we are talking much about precision medicine, and I think without real world data to support this is going nowhere. We need to understand the response pattern and disease characteristics in these small patient subgroups, and we need large data amounts for that. We also need real world data when we want to create new disease classifications, when we want to discover new molecular subtypes. And then also one critical thing is that we need to understand for example, the outcomes of multiple treatment decisions over time. This is not typically examined in randomized trials. You look at one treatment decision, one treatment, but I think more and more patients, as you all know, will be going through so many treatments over their life. And maybe there is a knowledge and wisdom also in what type of sequence they get. Like, what do you start with? What do you end with? Maybe this doesn't matter anymore. One thing that has been a key use of real-world data in Denmark is quality monitoring and treatment effectiveness. So actually, many of our data in Denmark, we collect a lot of data routinely on all inhabitants. They are actually meant for quality monitoring in hospitals to ensure that outcomes of treatments in lymphoma is the same in the hospital I work in as it is in another hospital. And if it's not, then you can look into what is the problem. Is there something we can improve? There's also so many questions you can address with these data that are important today, because we don't have the same healthcare structures around the world. We organize differently. Some areas, some countries are very much into centralization. Some countries may be more into decentralization, and so on and so on. And we can actually use the data we collect to see what kind of healthcare structure may be best for our patients. Also to look at the equality in outcomes. For example, if you have patients of lower socioeconomic status, do they have the same options, same treatment options, same outcomes as patients that are better off? Probably not, but this is important. And this is what we can learn from real-world data, especially because these patients of low socioeconomic outcomes also typically do not enter clinical trials, at least not in the same numbers in Europe. And then the effectiveness of new therapies, as was also said before this presentation, that this is really important to understand. When we introduce new, very expensive drugs, do they work at all, or do they work as good as we thought they would? Is it worth the money? And then there's so many questions that really will never be addressed in randomized trials. There's so many questions where there are no economical incentive to do a trial. And if a pharma company doesn't have an economic incentive to do it, then often it's not done. And randomized trials in academic setting is getting increasingly complicated now because of cost and organizational difficulties. And there are also several situations where trials are simply not feasible because of rarity. I think what we are seeing now is also a very good thing, that the use of real data, we're beginning to talk about it, how to use it, how to be structured around it, because maybe 10 years ago, this was a very different situation. This was really the Wild West. I would say that you could do whatever you wanted and analyze it how you wanted, but there's becoming increasingly focus on transparency and reproducibility. And we have a number of guidelines just to show that this is actually becoming an area of interest. We have academic guidelines like the START real-world evidence, how to plan and report real-world evidence studies. We also have regulatory agencies, EMA and FDA, guiding companies on how to analyze real-world data and what is really required to trust results that come out of a real-world data study. This is also another paper from a couple of years ago that also looked upon how to improve reproducibility and validity of these registry studies. And this is just to show that there are so many selections in a real-world data study that we have to be aware of, because every single time we do a selection, then there is a risk that results will be different. And this is not because necessarily the analysis are wrong. This is simply just because you do selections that impact the data and impact the results that you get. You do selections. Do you see my cursor? Can I ask? Okay. You do selections on the data on the data source. This is the first selection. You also do selections on when you have the data sources, how you combine it or which variables, then eventually you can end up with many different analytic data sets, despite you asking the same questions. And then, as you also know, then when you analyze the data, there's several ways of doing your statistical testing or grouping and things like this. So you can actually get different results from the same question, they more or less the same data set. So this is also something I will be talking about later. This is what I'll be spending most of the time on, the real-world data and development of new treatments. This is a largely unexplored territory, but it's really becoming increasingly interesting for many reasons. I think we can divide it in different steps, really going from supportive to decisive in drug development. If you look at the very early development efforts, like identifying gaps, unmet needs, or benchmarking of early phase 1 clinical trial data and things like this, then you can use real-world data, and it is certainly also used. And the thing here is that in the end, the clinical decision will not be based on real-world data. So you may have poor data, you may do your analysis in the wrong way or poor analysis, but what the result will be in that step is that you may have a research program that go in the wrong directions. I think at a later time point, when you use real-world data, for example, for benchmarking approvals, benchmarking single-arm trials, or maybe even as some will be discussing in future, I think, and it's more and more discussed, replacing control arms with the real-world data, then I think we have to be really much more careful, because in that situation, we can actually end up having drugs available on market that are approved, at least supported by real-world data. And if we're not very stringent on the analysis of these data, then we may have actually drugs on market that eventually does no good to patients, may even harm patients. So you know that the gaps in benchmarking, this is I think this is the simplest way of using real-world data in development of new therapies, and this is really to look for patients where there is an unmet need. And as you all know, follicular lymphoma is getting a more and more difficult disease to study, because the outcomes are simply so good. So it's difficult to improve the outcomes now, at least in the first line, except for some rare or maybe I would say less frequent types of high-risk disease. And I think one of the most clear high-risk disease in follicular lymphoma is patients that progress early within the first 24 months. And this is around 20% of the patients, but you see that the blue curve patients are really doing poorly, right? So this is a way of identifying an unmet need and really where to target development. We also have an abstract on ASH this year coming up, and I show you some of the results that are published now on the ASH website. This is a study looking at over more than 6,000 patients treated with frontline immunochemotherapy for follicular lymphoma. And you see in the different time eras, outcomes are a little better looking at the EFS in the time era 2010 to 18 compared to 2002 to 2009. So a slight improvement over time in EFS, but overall survival is really completely the same. So despite that we have so many new treatments for follicular lymphoma, actually it looks like that the survival of patients are identical in a 20-year time period. And this is also important to know. I'll talk a little about estimation of treatment effects, because I think this is really an exciting topic, and I think this is really important. The first question I would say is, can we replace the randomized controlled trials? When we discuss randomized controlled trials, often we have the randomization as being a key feature and really what makes it a randomized controlled trial. But there's actually a lot of other elements that we need to be aware of that makes it a trial. First of all, there is the structured assessment, right? That you have two treatment interventions and you assess patients exactly the same time. You assess them for progression at the same time point, you report the same way. And this is a huge difference compared to real-world data, where especially in studies where you use real-world data to compare different treatment outcomes. this can be a huge source of bias. You have the randomization, of course, to reduce the imbalance of risks like confounders or whatever. This is also something we don't have in randomized. Something we don't have in real world data often. But what's really the difference here is that you have inclusion and exclusion criteria that makes it the randomized trial and that characterize the patients that go into a randomized trials. And if you look at the pictures here, you will see also that this is all persons looking very much alike. They are almost similar. One is more tired, one is more unshaved, but they have the same dimensions. And this is really critical in a randomized trials, because all patients can get can get both treatments right. And I also showed here that when you have randomized trials, you have a randomization, and then you can ensure that you balance patients, you balance different patients with different characteristics. Also, the measurements you don't know, measurements you wouldn't know. For example, you can have patients that just by virtue have a shorter life because of comorbidity. These will be balanced. Whereas if you have a randomized, a single arm trial and compared it to external real world data, then you have no control of this imbalances. There are so many imbalances that you don't know of that you cannot adjust for. And you can just by virtue, and this is probably also happening many times, that you have patients that simply just do better because of some patient characteristics that are not really measurable in the trial setting, and then you compare them to external controls where you have a variety of different patients and patients that would never be considered for a clinical trial. And then you have a bias. And I think this is important to be aware of, because I think that with most new therapies, then we also know that sometimes the effect is quite marginal. It's not a major effect. So even these small differences in patient characteristics can actually mimic maybe a treatment effect that is not there. I want to show this by showing you a study from Denmark. This is not in lymphoma, this is in AML, but this study showed that trial participation actually predicted outcomes in AML, and if you look here, you'll see that this is the different age groups, that actually a very high proportion of patients entering clinical trials in Denmark. So we have 60% of patients below 25 years and 60% for patients above 75 years. So it's really a high inclusion rate. It's more than half of patients going into trials in the AML trials. And this is because we participate in these MRC trials, and then they are really kind of, I would say, less restrictive in terms of inclusion criteria. We include a lot of patients in these trials, but also many of the trials have been negative, right? So we wouldn't expect a better outcome just by being included in the trial, and many patients will also go into some kind of standard arm. But despite this, we see that there's a quite huge difference in the outcomes of patients enrolled in the trial, the red curve, compared to those who are treated outside the trials. And this is even when we have such a large proportion of our patients going into the trial. And this clearly shows that there are differences. There can be differences in patients, but when we adjust for the imbalances, at least the ones we can adjust for, so there's, for example, in performance status and things like this, the different remains. But there are also other differences when you compare trials to real world data, because, as you know, maybe the trial patients are treated at different centers. Maybe they have a different responsible physician. Maybe they have the AML expert that knows the trial very well and all the other treatments available. Maybe the patients off trial do not have the expert, and so on and so on. Maybe the research nurse takes better care of patients simply than the patients outside trials, but there are no dedicated research nurses and definitely much less resources. So there's so many elements that can make this difference. There are also other interesting studies on this in lymphoma, and I want to highlight a study out of the Mayo Clinic. maybe around a year ago, that looked at the trial eligibility criteria, especially the organ function criteria, typically seen in trials, for example, that they have to have adequate liver, kidney function, and so on and so on. You'll see here that patients treated with R-CHOP that fulfills the organ eligibility criteria of this trial, the Indian trial, actually they do much better, right? The blue curve is the ones who fulfill. The red curve is the patients who would be ineligible because they don't fulfill it. And there's a huge difference in outcome. And actually the difference here is typically much bigger than what is seen when you compare a new treatment to a standard treatment, for example. This is a much greater outcome difference. And when you look here, you also see that in the red curve, this shows what are they actually dying from, the patients that are ineligible. And you see here that they actually, the organ function criteria and the non-fulfillment of organ function criteria actually also leads to a much higher cumulative incidence of lymphoma death. So these inclusion criteria have a huge implication on how we interpret the results. This is also something we looked at in our Danish data, and we just, this is also being presented at ASH this year. You'll see here that the patients that are eligible, also using the organ criteria, the blue curves, they actually do much better than patients that are ineligible. But when you look at because there's also other differences between these patients, there can be differences in IPI and so on. So when we adjust for this further, there's still a quite substantial outcome difference. But what I really think is interesting here is we used the Goya trial, but we also used a lot of other trials, is that when you look at the stipulated line, this is actually the outcome of the control arm in the Goya trial. So when we apply our selection criteria to real-world patients and compare with how did the trial patients do, then we see a minor difference, but it's not as big as we expected. Actually, this means that we can more or less reproduce the outcome of clinical trial patients by applying the same selection criteria, at least in a healthcare system like Denmark that is very small and homogeneous. I would say something else that we need to be aware of is the time from diagnosis till treatment, which is also a major determining outcome of patients with lymphoma. We know that patients that needs treatment very quickly actually do much worse. So these patients that have 0 to 6 days between diagnosis and treatment are clearly the patients doing worse, whereas patients that have a long time do much better. And this is not the same as recommending that you wait a long time with treatment. This is of course because patients who need treatment early or who are treated early are very sick. And these patients are also patients that would typically not be in a trial, because just enrolling patients in the trial, informing them, having them to consent and doing maybe a centralized review of pathology will take more than a week. So these are the high-risk patients that are left behind as well. One of the things we have to be very aware of when we look at real-world data and compare these data to other outcomes, or compare or just describe the outcomes of real-world data patients, is the time zero problem. And this is really something we need to be very careful about when we describe the outcomes and when we measure survival. When do we start the clock? When do we start the time? Because in real-world data studies, we need to be sure that we start the time after, for example, we have assessed the eligibility for this study we want to do, and also after we have had the intervention. This is marked with the A, the intervention and the eligibility. If the eligibility assessment and the intervention is before you start the time, then you have no immortal bias. If the eligibility is on the date you start the time, you have no immortal bias. But if you start the time, for example, at the diagnosis, the time of diagnosis of lymphoma, but your treatment interventions you want to study is something after diagnosis, then you have a problem. And this is something you often see, for example, in transplantation trials, that you have a plateau the first 6 months in the survival curve. And this is because there will be an immortal bias, right? You cannot, if you start the clock and you require that the patient had to be transplanted, but you start the clock at diagnosis and transplantation only occurs 6 months after diagnosis, then you will create an immortal bias. And this is very difficult to interpret when you compare across trials or across studies. And we have other kinds of immortal bias. Something I've been working quite a lot on with colleagues is the use of high-dose methotrexate. This is a typical immortal bias for preventing CNS relapse, because if you give high-dose methotrexate after R-CHOP, for example, after completing 6 cycles of R-CHOP, but you look at patients from the start, from this date of diagnosis, then all CNS relapses that occur before you end your R-CHOP therapy will not have had high-dose methotrexate, but it will appear as it was the effect of high-dose methotrexate. And I'll show that here. This is also something we'll be presenting at ASH. This is almost more than 2,000 patients with treated with high-dose methotrexate. This is the yellow curve, or no high-dose methotrexate. This is the dark curve, or the black curve. And these are all high-risk patients for CNS relapse. So if you look at the time from diagnosis, you'll see that it appears a little like there is a slight reduction, at least at some time points, when you use high-dose methotrexate. But when you look at this as the time of end of R-CHOP, then you see that this difference really disappears. There's no protection using high-dose methotrexate. So, but this is of course because you will introduce some kind of immortal bias or guarantee time bias when you use it. When you analyze high-dose methotrexate and then look at the time from time 0, the time of diagnosis, and not the time after R-CHOP, where all patients could have had high-dose methotrexate. This is another thing I think also can hamper the comparison when we look at real-world data and clinical trial data and expect the same outcomes. This is especially for progression-free survival. You know that clinical trial settings typically have like very scheduled follow-up with sometimes regular CT scans every 3 months, 6 months. This is something we, at least in the Nordics, doesn't do that much in the real world setting. We typically monitor patients just clinically. But if you have, like, for example, 2 patients, this is illustrated by the 2 red lines. This is 2 patients, they have a relapse or progressive disease at the same time. And if you do regular assessments all the time, then you will capture it next time you do a CT scan in the trial patients or in the real world setting where you use CT scans. Whereas if you compare to a setting where you just follow patients clinically, then you have to wait until patients appear with symptoms. But if you look at the survival after the relapse, then the relapse will occur here and will be registered here in the patients in the real world setting, whereas it will be registered here in the clinical trial setting. And of course, this can make a huge difference and give some kind of lead time bias in the trial patients, where you simply just diagnose relapses much earlier. And I think this is also why you have to be very careful when selecting the endpoint in clinical in real world trials or real world studies. Overall survival is very unbiased, and time to next treatment may be also a little less biased, but things like progression-free survival can be really heavily influenced on how you measure progression and how often you assess for it. There's a nice study. This is not in hematology, but this is in as I recall, it was oncology in general. But this is looking at patients. It's in JCO from 2019. This is looking at studies that were done in the randomized clinical trial settings, but where they also had studies in the real world setting afterwards of the same interventions. And when you look at here, the curves here, the red curve, here the stipulated red curve, this is the best fit line between results seen in a real world setting and results reported in a randomized trial setting of the same intervention. If this was completely the same, it should be the blue curve, but this is actually the red curve. And this is a really poor alignment. So you get often completely different results when you go in the real world setting compared to the trials. And when you look here, the red dots actually signal where the hazard ratio of the observational study, the real world study, it's not even within the 95% confidence interval of the randomized trial. So it's really completely different results. And when you look at different adjustments and studies that were done, like the overall number of studies, but also studies that they thought was very well matched, the real world data and the randomized trial looks very much alike. then there's no agreement. There's only agreement in 40% of the cases. So we shouldn't expect necessarily to be able to reproduce outcomes of clinical trials consistently in the real world setting. And of course, the question is then, is it the drug that doesn't work as we thought, or is it because we select patients differently? And it's probably a mix of these things we have to be aware of. CAR therapy, cellular therapies is another situation where you have single-arm trials, and we have single-arm trials with really amazing results. And then we have real-world data. This was the SCHOLAR-1 study, where we use this as benchmark and see that when we use CAR therapy in patients with relapsed refractory DLBCL, then the outcomes are really much better than what is seen in the real world setting, where the median survival was, I think it was 6 months only. And these studies were done quite nicely, and they were also using more or less same inclusion criteria, same definitions of refractoriness, that you had to have a refractory disease or relapse within 12 months and so on, and same degree of poor health, measuring with ECOG performance status, for example, or comorbidities. But we have to be aware that even though we have these dichotomous variables that we think we match on, like saying you're refractory and you fulfill the definition of refractory, and this is the definition we use in the real world setting and the trial data, then there are, of course, very different degrees of refractoriness within this within these definitions. You can have patients that are very refractory, like patients that for example, never respond to therapy, or you can have patients that are refractory because they have a very slow relapse 3 months after ending therapy. And these patients are very different, but they will both be classified as refractory. And patients that have a very slow relapse are probably more likely to go into a CAR trial, whereas patients that never respond to anything and die very quickly, of course, never go into a CAR trial, or at least a lot less likely. And the same for ECOG performance, for example. That can be, even though you have ECOG performance 0 to 1, and this is categorical variables, then you can be ECOG performance 0 to 1 for different reasons. If you are ECOG performance 0 to 1 because you are sick from your lymphoma and it resolves after you start treating, or if you're ECOG performance 0 to 1 because you had or maybe ECOG performance 2 because you have a previous myocardial infection or cardiac failure, then this is a very different situation. And again, the patients with the severe comorbidities leading to poor performance are probably less likely to go into a trial compared to patients that have poor performance because they're sick from lymphoma and respond very fast. And I think there's something to this. When you look at the real CAR experience from England, then you see that patients who get infused actually do very well. They have an amazing survival and the results are really good. Patients who don't get infused are actually doing quite poorly. And this is the overall survival. This is not so unexpected. Of course, the treatment works, and patients who don't get infused are probably more sick. When you look at the event-free survival after infusion of CAR, you see that it's not as great, right? If you look at the overall survival curve, the red here, and the same patients with their event-free survival, it seems like you have quite a lot of progressions after CAR, but nevertheless, they managed to keep them alive. And this is something that also suggests that probably these patients are a little different, right? They are different in terms of maybe tolerating next lines of treatment, maybe easier to get them to an allogenic as well, and things like this. This is an important bias. But nevertheless, I want to say that, of course, CAR therapies are highly effective. We know that, and we also would recommend you to go to the ASH website now and look at the result. It really is amazing results they have when they are randomized against high-dose therapy in the second line setting in DLBCL. I want to spend a little time on discussing how you should look, or how you can look upon survival benefits observed in observational studies, and especially in the special situations where you have a negative randomized control trials. And one of the things I have been working a little on is the use of rituximab maintenance after treatment for follicular lymphoma in first line. And as you all know, the PRIMA study was positive in terms of PFS, but when you look at OS, even with long follow-up, there's no survival benefit of giving rituximab maintenance in the first-line setting. And actually, in Denmark, many have stopped using it because it's just the same as treating continuously to avoid relapse, where you could also say to the patients, you can have a break in your treatment and you can come back when it is active again, and then we restart treatment. You can even restart rituximab single agent at that time, right? So, but of course, there's been done a lot of real-world evidence studies looking at the use of rituximab maintenance as well after this was introduced. And one study was out of Denmark, where you see here the all patients in the CHOP-CHOP-like treated patients. The blue curve is patients who got rituximab maintenance. The red curve is patients who didn't get maintenance. And if you look at the PFS curves down here, it looks maybe a little better than the original clinical trial, but nevertheless there is a PFS advantage. But when you go and look at the overall survival, the top curves, then it's really a quite substantial overall survival benefit you see. And this is special because typically you don't see a PFS benefit of this magnitude translating into an OS benefit of this magnitude in follicular lymphoma. And this OS benefit actually remained after different adjustments for differences in FLIPI and things like this. But it's probably a sign that there is some residual confounding that we are not capturing. And what you can ask yourself is, for example, if patients have dementia or patients have a concurrent cancer, like diagnosed with a colon cancer because you have a residual FTG avid area after treatment with immunochemotherapy, and you biopsy them and they have a colon cancer or they have something else that happens, of course, and this is not so rare, then would you then give these patients rituximab maintenance? Probably not. So there are some selection bias in this, or at least potential. And actually, we did a similar study also in Danish data a couple of years before this study. We didn't use the registries. The previous study I showed was registry-based, but we didn't use the registries. We used the health records. We looked at actually detailed patient outcomes in 3 hospitals, not nationwide. And we found that there was no substantial difference in PFS, time to progression or time to next treatment. But when we look at the OS, overall survival here, we actually also identified that there was an advantage of giving rituximab maintenance. This is, of course, again, a little odd, especially when you see no advantage in PFS. And we actually found that this was because there were so many in the rituximab, or the group that didn't get rituximab maintenance, that died from other causes. 5 of 7 that died within the first 2 years after FL diagnosis, they were actually unrelated to lymphoma. So you had death from lung cancer, from COPD, from myocardial infection, from unknown, just died outside hospital. And of course, these patients, it's difficult to imagine that, for example, it was the rituximab that maintenance that led to reduction in these kind of deaths in the rituximab arm. So there's clearly some unmeasured confounding here. And this is one of the problems when we look at outcome studies in real world data, that we cannot really control the exposure, right? So we have a group of patients that get a treatment, and we have a group that didn't get a treatment, but we cannot, like a clinical trial, assign the patients who originally didn't get the treatment to the treatment, because we want to be sure that they are completely equally likely to have the same treatment, right? So there is already a clear selection before you look at the treatments, and patients have already been assigned to a treatment in the real world setting, and often there will be a reason for different assignments to different treatments, and these reasons we cannot always measure. There is a methodology in real world data studies or in epidemiology that is called negative control outcomes. This is quite complex, but I'll try to show you how this works. If you have, for example, your intervention that would be rituximab maintenance, and you want to measure this effect on overall survival, then of course, there are some biases, like you can have age performance score that may affect the likelihood of being prescribed rituximab maintenance, but may also in itself affect the likelihood of surviving. And you can adjust for these things if you can measure them. But sometimes these biases that are actually related to some other biases. This could be, for example, poor performance could be related to smoking or unhealthy lifestyle, or something that you cannot adjust for because you don't have the data maybe. And this unhealthy lifestyle or this correlation, this may also influence whether you would like to prescribe rituximab maintenance. It may influence overall survival, of course, but it may also lead to completely different outcomes. And this is these associations we want to be sure that doesn't exist, like you don't have a situation where you measure the rituximab maintenance or any other therapy given for hematology cancer to have an effect on an outcome that is completely unexpected. So we did a little more to look at this, and this is unpublished data, but it's also from the Danish registries. And when we just look crude on overall survival for patients treated with maintenance and no maintenance, you see that the red curve is really better. They just survive better, patients who get maintenance. And this is in contrast to the clinical study. And actually often we have the different problem, right? We have the problem we show differences in clinical studies, but no difference in real world data. But here we have the opposite direction. But what's strange is that when you look at different time errors, even time errors before you even use rituximab maintenance, like the blue curve from 2000 to 2005 or 2006 to 2010, also before this, there's no difference in overall survival. It's all the same. So you cannot measure the use of rituximab maintenance, at least on overall survival, even though you see that there seems to be a benefit for patients assigned to rituximab maintenance, and they were mostly treated with rituximab maintenance in the late era. But when you look, for example, at the risk of having dementia or any other common cancer for patients who complete immunochemotherapy, then you see that the blue curve, this is patients who didn't get rituximab maintenance. They are actually more likely to have dementia or another cancer as well. And it starts right after the therapy, right after the immunochemotherapy. So this suggests that there is some selection here that may influence survival that we do not control in our studies. And this is, of course, not something that we believe rituximab maintenance has an effect on. Like, this wouldn't have an effect on breast cancer, GI cancer, or dementia, for example, things like this. So this is suggesting that there are something we do not control, and we cannot really completely trust this overall survival benefit we see. Really, then I would also say that real world data and randomizations are no contraindications. So, contradictions. There are really many ways to use real world data or real world setting in a clinical trial, so that we get the best from both worlds. You can have your patient pool, your homogeneous patient pool characterized by simple inclusion-exclusion criteria. You can randomize them. All these things are like a like a randomized trial. But then when you go into the intervention or the data collection, you can use more real world settings. You can, for example, if it's a very simple intervention, you can probably do this without too many specifications in the trial. You can maybe say that you can do as you usually do, and not like a very detailed list on what you have to do. And if you have a very hard endpoint, for example, follow up for overall survival, then maybe you can use, for example, data collected anyway as in routine as your outcome measure, and not necessarily your CRF from a trial. So you can actually mix this, and this is getting more and more normal. And I want to highlight this. This was done in Sweden and was published very well. And this is not in hematology, but this is in myocardial infarction, and looking at the use of oxygen, like should you give patients oxygen or not when they have a myocardial infarction. And this study actually just used very simple inclusion-exclusion criteria, and patients were randomized using the Sweetheart. This is a registry, so they used their own patient registry to do the randomization. Then they did the simple intervention, giving oxygen or giving no oxygen, very simple. And then they had the primary endpoint, which was all-cause mortality, which is captured routinely in the Nordic countries because we all have a personal identification number, and if you die, then this is like captured everywhere because this is also what's used for tax or governmental support or whatever, education programs, everything. So you cannot avoid being registered as dead if you are not dead, or if you died in Denmark, then it's very, very difficult to imagine that you are not captured in these registries. So you have a very safe outcome capture. And this was published very well, and it was really nice because this is actually a type of study that use randomization, so we avoid the confounding that we cannot control because we don't know everything about the patients. It used less selected patients, right? It had a very wide inclusion criteria. They can include a large number of patients. They have simple interventions. They are inexpensive. This is actually the type of trials we can conduct in academia. But of course, the data quality in these trials will be more variable. And what we really miss in a trial like this, and what also limits its use, at least in some situations with hematology, is that the safety data is, of course, very it's weak, because if you don't have regular safety assessments, there will be so many AEs that you will not capture. So you can imagine that this will be a very difficult setup to use for a new treatment. For example, a new treatment for CML, as the quiz was about, because you do not capture nausea, you do not capture fatigue, you do not capture all these important things as well if you just use a registry for survival. Yeah, I'll just touch very briefly on pharmacovigilance, and then I will end the presentation. I hope we can have some discussions, because this is the more classical use of real-world data for pharmacovigilance or for assessing late toxicities. And we really have a strength in hematology because we have, when we use real-world data, because we have, first of all, small patient subgroups, then we have sometimes some rare but very important side effects. And then also, because many of our patients are luckily now surviving for a long time, then we need to be much more interested in having a good data on the late adverse events, and not just adverse events that occurs during the conduct of a trial, a typical trial of 2 to 3 years maybe. And I want to show an example of this. This is the RCHOP trial from the US RCHOP trial published by Haberman in 2006, where we really see that RCHOP compared to CHOP is, of course, much, or at least somewhat better than CHOP alone, in terms of PFS and OS. And this is, of course, very, very old news. Maybe we have a new treatment soon with the Polaris trial, who knows? But what was shown in this trial was that there were similar rates of infections. If you have rituximab or no rituximab, you still had a 65% risk of any grade infections. And even actually, when you look at the grade 3 or 4, then it seemed to be that CHOP-treated patients actually had numerically at least a little higher infection rate. But I would say there were very rare but very serious adverse events that were not captured in the early rituximab study, that just showed that it had an overall survival benefit. And this is, for example, the multifocal leukoencephalopathy that was captured only in the post-marketing setting, only after we had the drug available. And this was shown by review of safety databases and other real-world data sources, where there was a nice publication presenting 57 cases of PML, mostly in lymphoma patients. And this is how we knew that this could be an issue. It's not a daily clinical problem at all, but it's something we have to be aware of in a very small minority of the patients, because it's more or less always fatal. But I think what is important from this learning is also that when you went back and reviewed cases, then 22 of these cases, almost half of them, were never reported to authorities. So we have a very rich experience in the real-world data setting. We have a very rich experience in our routine clinical setting that we do not report to authorities, so we can warn on these things. Maybe because we don't have good ways of capturing this experience. We don't have good automatic ways using our real-world data to learn these things. So we have to go back more or less manually when we get an interest in a specific side effect. And this is, of course, time consuming, and you have to have first the signal or the idea if there is a problem to do this at all. But I hope in future, maybe with more electronic health records, with more structured data, that we will actually be much better at using real-world data also routinely for pharmacovigilance. This is another example of this. If you look at the MINT trial, sorry, this is the Recover 60 trial. You look at patients got 6 CHOP, 8 CHOP, 6 R-CHOP, 8 R-CHOP. When you look at the cardiac function, it's more or less the same. There were no major difference in cardiac AEs. And of course, we also have this old learning that if you give more doxorubicin than 550 milligram per square meter, then you increase the risk of heart failure. But when we give 8 cycles of R-CHOP, then it's a maximum of 400. So we are well below the threshold for heart failure. We looked at this in our Danish registry data. We can capture all lymphoma patients. We actually register them with treatment in our lymphoma registry. We can also capture all cases of cardiotoxicity, because if they go to a hospital, a public hospital, and all hospitals in Denmark managing severe cardiac disease, or at least like 95% of patients will go to a public hospital. So we know that if patients that had lymphoma later encounter, or if we know if they are later seen at a cardiology department, or if they have a diagnosis of heart failure or myocardial infarction or something like this. So we try to look at this and correlate this to the number of treatment cycles. And when you look at this in the real data, then you really see that patients who had more than 6 cycles of R-CHOP have a substantially higher risk over time for getting a diagnosis of heart failure. And this is not just a surveillance bias because they are lymphoma patients and they go to doctors more often and they get their diagnosis, because if you look at patients who get no anthracyclines, it could be CVP or bendamustine, then they have a much lower rate. But even patients who get less anthracycline have a substantial rate. But really, this is an argument for not going beyond the 6 cycles, also because there is no really good evidence that 8 cycles is better. But this is again something you can measure in real world data. Just want to advertise a little on that we have a policy paper from 2018, also containing a section on real world data and how to use this in future pharmacovigilance. And I expect also we have an update of this policy paper coming out soon, where we discuss some of the problems using real world data, as you all know, especially the problems of EHR systems that don't speak together. Like you can have in one hospital one EHR system and the other hospital have a completely different, even within Denmark, and we cannot exchange data. We cannot really leverage the possibility of looking at data in big scale simply because of technical issues now. Yeah, actually, I think I would like to stop here, because I think I already said a lot and maybe also use my time. The last part is not as important, I would say. So maybe we can stop here and then have discussions on these topics at one point, or questions. Would that be okay?
Yeah. Thank you. Thank you so much, Professor Tarek. That was an eye-opener, I would say. So many things to learn from what you spoke. We have a lot of Indian faculty here who will be itching to discuss with you. We'll just take a 5 minutes break to give the answer of the quiz to the postgraduates, and then we will start with the question answer session.
Okay.
Just share my screen for that. So friends, this was the Sunday quiz for you. It was related to the drug asciminib, and you were given 7 statements, one of which was wrong. For some of you, asciminib may be a new name. So just to introduce you, this particular compound, asciminib is a chronic myeloid leukemia drug. It is available in the market as Samblix from the house of Novartis in 2 strengths, 20 milligram and 40 milligram tablets. That's its structure. And that's Novartis pack of asciminib 40 milligram tablet containing 60 tablets. And this is relevant because just a week ago, October 29th this year, the US FDA approved this particular compound, which has a very novel mechanism of action for the patients of chronic myeloid leukemia. So with this approval, I thought this would be very relevant to discuss this compound. This is the ABL kinase, and you got the ATP binding site where exactly your usual TKIs work. So imatinib, nilotinib, dasatinib, ponatinib, all these work at the ATP binding site. While as far as this particular compound is concerned, this works at this pocket that is called myristoyl pocket. So it's entirely different mechanism of action. And that's the reason why, one, those who got resistance to all this ATP binding TKI, this drug is effective. And second, you can combine these drugs. A number of trials are on to combine asciminib with any one of these, imatinib to ponatinib, all of these. That's the rimmer diagram showing you once again. This is the site where nilotinib acts, and this is the site where asciminib acts. So these are 2 entirely different sites. These were the 7 statements made. So asciminib is an oral. Yes, it is an oral compound. It is ABL inhibitor. It is ABL inhibitor and specifically targets the particular pocket, myristoyl pocket. So this was a correct statement. It is licensed as a monotherapy for patients of chronic myeloid leukemia who have received at least 2 TKIs. So maybe imatinib followed by either nilotinib or dasatinib, or maybe even all of them, etc., etc. Or those who have developed 315I mutation, who have very limited options, usually just ponatinib or transplantation, etc. So this is a correct statement. Then assemble trial was the registration trial. That part is correct. And this was carried out in CML CP patients after 2 or more TKIs. We'll come back to this statement a little later on. This tablet is marketed as a film-coated tablet, correct. Samblix is the brand name, correct. The dose is 40 milligram twice a day, or it can be used even 80 milligram once a day. Both have been US FDA approved for CML chronic phase, while the dose is very high, 200 milligram twice a day, total of 400 milligram for patients who have got T315I mutation, which just tells you that there is no dose limiting toxicity when you're using 40 milligram BD or 80 milligram OD. And like nilotinib, it is best taken on empty stomach. So that part is correct. 2 hours before you should be fasting, and 1 hour after the tablet it should not take. And therefore 80 milligram OD may be more practical than 40 milligram BD. Unlike the various TKIs that we have been using for CML patients, this particular compound does have almost no off-target activity, as a result of which its safety profile is tremendous. And especially all the TKIs that we use have some cardiac toxicity, some more, some less. This one is almost free from the cardiac toxicity. And it is also correct that even if you have got severe hepatic and renal dysfunction, you do not need dose adjustment for asciminib. And lastly, the research is on combining this with imatinib, nilotinib, ponatinib, etc., because they act at the ATP binding site, and asciminib is working at a different site, that is myristoyl pocket. Therefore, these combinations are effective against compound mutations, and it also permits you to decrease the dose of ponatinib. You are aware that ponatinib cardiac toxicity is dose related. 45 milligram is more than 30, 30 is more than 15. So when you combine them, you can use lesser dose of ponatinib, and that's the principle on which this research is going on. So all look quite all right. That what is wrong? The wrong is that the assembled trial was asciminib versus botulinib. and not ponatinib. And usually you use the second line treatment as nilotinib or dasatinib. So many of us will use botulinum for the third line, and that was compared with asciminib. So ponatinib was wrong and bosutinib was the correct answer. So this statement was wrong. You were supposed to reply as 3. This is a publication 2 years ago, New England Journal, 2019, asciminib in chronic myeloid leukemia after failure of 2 or more lines of ABL kinase inhibitor. And this is an article by Coates and his group, third line therapy for chronic myeloid leukemia, current status in 2021 and future directions. This is a very good article for postgraduates to read, because after you have used 2 lines of therapies, imatinib and either dasatinib or nilotinib, the success of TKIs after that is very poor. You don't have many options, even if you do not have 315 inhibitor. And this is safety and efficacy of asciminib treatment in CML in real life practice. And we just heard how research and real life could be different. So this is an Italian article about the real life use of asciminib. So the answer that was expected from you was 3. Well, I'm afraid there are not many correct answers. Many of you have given answer 4 or 5 or 6. These are total 3 correct answers. 2 of them were by Dr. Virtisha, Dr. Amit Khurana. Congratulations to both of you, but you were not the fastest finger fast. The winner has to be the fastest finger fast, and that was once again Dr. Akanksha Garg. Congratulations to her. She has developed a habit of winning the prize. So Dr. Akanksha Garg, tremendous knowledge and the speed with which you answer. As you know, she is Assistant Professor, Department of Medical Oncology at GCR Ahmedabad. So once again, congratulations to you, and your prize will reach you. Thank you very much. And now we go to the question and answer session. I request all the discussants to open their videos, and we have an active discussion with everybody. Till you raise your hand, may I ask one question which is asked by the audience? Dr. Kannan has put up a question to you, Dr. Tarek. Can you give an example where a randomized controlled trial has shown a benefit for a particular drug, but the real world data was at its variance?
Yeah, actually, I saw this question. I was thinking, and I did, nothing really came on top of my mind in this setting. I think the other way around, we have seen quite a lot. And then I was thinking on the current debate that in the FDA now, where they have granted quite a lot of accelerated approvals, not in hematology, but in other oncology situations, where they are actually reviewing them now, because they were done on single-arm trials, but then after this, the randomized trial maybe was never done, or maybe was negative. So you can at least have situation, at least, as you know, when you have really, really nice single-arm trial data that you do a randomized trial afterwards, and then this is negative. That's plenty of situations of that, but I didn't just came across this example that was asked for. Maybe some of India faculty will have another example. I don't know.
Okay, so we'll put this question to the Indian faculty, and whenever you are ready with an answer, you can do that. Meanwhile, you can ask questions to Dr. Tarek or share your experience with it. Anybody would like to start the ball rolling? So here we have Dr. Joseph John. Go ahead, Professor.
Thank you so much for the talk. I had put one in the chat box. What is the consenting process which you do? So considering that you're doing a retrospective analysis, so what I understand is that it could be a prospectively planned study or a non-prospectively planned study, but you are doing a retrospective analysis of the real world data by after the mining. So what's the consenting process which you do for both these?
That's a very good question, and I think there's no single answer to this, because I think what has been shown in the last couple of years with more interest in these kind of studies is that the regulations in different countries are very, very different. So in Denmark, if you do a prospective study and enroll patients, like if I say I would like to do a cohort study of patients treated with ABVD in Hodgkin, and maybe even do extra assessments on them, but maybe just do a cohort study, then in a prospective fashion, then I would have to consent them, of course. If you do a retrospective study of clinical care that did happen at one point, then as a rule, you would need to consent them as well, but often you would get an exception to this because it is not feasible, because if you want to study DLBCL, for example, then some patients would have died, and maybe sometimes getting consent would be so complex that you would not do the study. And then in Denmark, we have a rule that says that if it's for the good of society and it has positive health implications for in general, then you can get an exemption from this consent. So this is where you look at health data. This is where you look at health data from health records or enroll patients in an observational trial. If you use registries alone, if you use the registries made for quality assessment and things like this, these do not require consent. These we are put in, all Danes are put in these, whether you like it or not. And to do studies on this, you would have to ask the data authorities, but no consent from the patients, because this is typically population-based, and we cannot go out and ask, for example, all lymphoma patients, and if we match to 20,000 controls, then this would be very difficult, or at least these studies would not be done. So for these studies, no consent or considerations of consent, but then you have to instead apply data authorities, and you have to live up to some very strict data regulations now, especially in Europe. So but I know this is very different in different countries, and when we submit papers sometimes to journals, then we get the questions like, did you have an ethical approval? And no, in Denmark we do not get an ethical approval, and the ethics will not even look at these studies, because this is not within the ethics to assess these retrospective studies. If they only use data, if you use tissue, this is another story. But if you only use data, then it's the government or it's the government that will eventually approve. Actually, it's the politicians now who approve that you access these data, and then it has to, of course, they have to see that what you do will have positive effects on healthcare in general, for example, for future patients. So this is how it's done in Denmark, but it's different everywhere in Europe.
Joseph, any comments?
So what I understand is that if it is a prospective study, you have to take it. If it is a retrospective study, but depending upon the situation, a waiver can be asked for.
Yes,
is there a role of blanket consenting at the time of admission, and therefore in future you can ask multiple questions and analyze the data?
Yeah, but this is completely true, but they don't give any consent to registries in Denmark, because our registries are, for example, used for hospital payments and things like this, but they're very detailed, but you cannot avoid being in them, because then there's no registration of activity in the hospitals in Denmark. So these are also the nationwide data we often use as for studies, and then for our lymphoma registry, for example, this is a quality registry. So you also have to, we are obliged as doctors in my hospital and in all other hospitals to put patients in this quality registry, because this is used for the main purpose of this registry. The secondary purpose or secondary use is research, but the main purpose is that we want to track quality. We want to be sure at all times that every hospital has same outcomes, that every hospital have same quality, because then if a hospital show with worse quality or worse outcome, then you can start an investigation. So this is actually the purpose of this registry, and it's mandatory for doctors to register patients. But I know this is very different from between countries. So it's maybe the Nordic countries have a unique situation there. It's more or less the same in Sweden. This is also why we have so nice registries. And there are other countries where you are much more reluctant to put people in registries, maybe because also of historical reasons and things like this. So there can be so many differences.
Thank you. Dr. Abhishek.
Sir, amazing lecture. I think you basically presented all the evidence we already know as clinicians, but we failed to acknowledge. Sir, I've been participating in a number of trials, and what I feel, you might correct, I might be wrong, selection bias is what puts these trials off. there is always a selection bias. You select the best of the patients for the clinical trials and they give you amazing results. So, but these election criteria are prepared by some of the best people from amongst us only. So you said you basically showed what is wrong with these trials, but what is the remedy to it?
That's a very good question, and thank you very much for that. I would say the solution to this, I think, is already very much on the way, because I think there's so much focus now on regulatory agencies to have more inclusive trials. So I think this will be different in the future, I hope. I think that the exclusion criteria in the trials, that's true that many of us are involved in setting them. They are actually fine in the way that you want to know exactly the effect of the drug, which I guess is the purpose of the trial, but you just have to be aware that you only document the effect in this exact population, of course, that you select. And then you may have very different effects when you take it out in a broader population. So this is also why I think it's fine to have a clinical trial that is very, very, like, it's not fine, but it's understandable that you want a clinical trial where you can assess the effect of the drug very precisely in a homogeneous population. But if you have done this, then it should be almost mandatory, in my view, to monitor the effect of a drug after you introduce it to market, to be sure that when you broaden it to a much wider population that doesn't fit all these very nice eligibility criteria that makes eventually makes a very fit population and almost much fitter than the average patient, then you have to assess it in the real world data afterwards. And this is also becoming more and more normal to ask for these post-marketing studies, or at least also from, for example, in Denmark, we are getting more and more interested in looking into the efficacy after approval, because as you also know, these drugs are very expensive, and we don't really know if we actually get what we are paying for when we take it into the real world setting, and we need to know that, of course. So,
can I follow it up? The reason why I'm asking you is the patients we have in our routine clinical practice in India are much less fitter than the ones which are being put into the clinical trial. So real life practice does not match what is being done in a trial. I'll just share I won't name the drug or the company. Real life experience with a myeloma drug post the clinical trial has been the disaster. So I wonder what the clinical trial did and what the real life data feels like. So basically, shouldn't the clinical trial criteria be the same as the real life criteria? You take any patient, fit, unfit, if he deserves the drug, he deserves the drug.
I fully agree with you. That would be the dream situation, but I think this is not only. I think this is not only the drug companies that put the inclusion and exclusion. I think also the regulatory agencies sometimes have a role to play in this. That they require this to be sure that the trial is very safe, that you don't expose patients to unnecessary harm or encounter some unexpected AEs. And of course, this is. The more healthy you are, the more likely it is that you don't have some serious effects. But of course, this is wrong in a way, because then we take it out after the trial to all the patients, and then we expose everyone to this, and this is not fair. But yeah, so I fully agree with you.
Abhishek, I would like to give you a fantastic example of chronic lymphocytic leukemia, alemtuzumab versus chlorambucil. And alemtuzumab gets approval for first-line therapy in CLL. So anyway, Dr. Govind Babu.
Yeah, so let me again echo the same thing, that it was a wonderful lecture, and a lot of things that we many times take for granted, you highlighted very well. My question is, in evidence-based medicine, we have categorized all the evidences that come from the clinical trials based on whether it's a meta-analysis or a randomized clinical trial. And accordingly, we give importance to that particular data. So do we have anything like that for real world data?
Do you mean like a pyramid for
yes,
for
exactly
real world data?
Yeah.
I'm not aware of that we have anything official like this, but of course there will be different qualities in real world data studies as well, depending on how you do your analysis, but also especially on the data sources, because If you have a prospective observational real-world data study and then you really control your measurements, then it's of course much better in many ways than if you do a retrospective study. And if you even are in a situation where you need to collect your data retrospectively, like if you have to assess outcomes retrospectively and all these things, because then you are really in the risk of a lot of bias. But I haven't I didn't come across like any pyramids like this, but there are different qualities of real world data studies for sure. And this is also, I think, what you see when you look at because there's so many real world data studies out, and there's so many studies studying the same topic, but you have sometimes very different conclusions. And I think to your point, then it gets quite important that we start to be more interested in the hierarchy of this and the quality of this, because at least you can say that not all of them can be true, right? So if you have different results in different directions, then maybe the truth is in between, but maybe also some of the type of studies are really biased and shouldn't be viewed as evidence at all.
Yeah, exactly. That is what my point was, because if we have the hierarchy as we have for the evidence-based data, if we have a similar scale here, then we would be better off adopting this for our practice. So that's what my point was. Now, the other question I have is, how do you integrate the evidence-based data that we have and the real-world data and try to make the best out of it for the patient sitting opposite to you?
I would say when we talk about treatment selections and treatment of patients, I think still the trial data weigh quite heavily. But there's so many other aspects when you sit with the patients that where the real world evidence matters a lot. For example, I can give you an example that fertility in Hodgkin, we have been looking at that problem and looking at beer cup and ABVD and what the problems are. And this is not something that was really well captured in studies, because typically studies in Hodgkin run for 5 years maybe, and patients get children after the 5 years or want to have family after the 5 years as well. And there we looked at the data in the Danish registries, and I've also looked at a Nordic level and seen that, for example, beer cup is associated with clearly much more infertility, especially in males. And data like this, and also like similar situations, I use in the discussion with patients, because I think it's important, because there's so many situations where we don't have a clear-cut answer in hematology. We don't have a clear right or wrong, right? There's nothing wrong with giving patients beer cup. There's also nothing wrong with giving patients ABD, but there may be other subtle aspects of this or other evidence that should be put into this, generated in the real world setting, that makes, in a discussion with patients more likely to choose what's right for them. This can be things like this, like what's the long-term side effects? Maybe for some patients, they would not at all accept a risk of infertility. Maybe some patients would accept it, but would more go for highest PFS if this is important, and things like this. So patients are so different. I think this is the way I use this. I use it quite a lot.
Thank you so much.
Thank you, Dr. Govind Babu. Dr. Chandran.
Thank you, sir, and thank you, Dr. Tarak. My question is, when we talk about retrospective analysis of prospectively collected data, does that perfectly fit into the so-called real-world evidence?
Yeah, I think this fits very well into real-world evidence, and this is how we do a lot of studies. And I think what is also, to the point of the quality in studies that we discussed before, is becoming more and more pertinent and also more acknowledged as being highly important, is that, like, when you do a trial, then it's also a good idea to do your protocol and your statistical analysis plan, and maybe even publish this on a web page before you start your analysis. Because when you actually have your data prospectively collected, and if you truly do your analysis plan without looking at the data and have a hypothesis, and one hypothesis, one primary endpoint, then you almost get the same rigorous analysis as you do in a prospective trial, a clinical trial. But the problem is sometimes, at least historically, and is that that many analysis have been very data driven, right? So you know the data, you know kind of how it looks, and you get a feeling that, oh, there's nothing here, but if you look at it like this, there's something there, and then when you do like this, then you end up, this is also okay, but then you just end up having to state really clearly that this is exploratory, and you need to confirm it because you lost all your alpha control in approaches like this.
And one more question. You were talking about rituximab maintenance in follicular lymphoma after upfront treatment. So you have contradicting evidences from randomized controlled trial, no OS benefit, but in real world practice, it has improved the overall survival. So in such situations, for a clinical practice point of view, how would you take it forward?
Yeah, so this was an example I gave to show that you shouldn't always believe in what you get out of real world data, right? Because of course the clinical trial was negative in terms of OS, but then you show in real world setting that there is an OS difference. And this is of course a very strange situation, and this is also an unexpected situation. But this was just to show you that if you do these kind of real world data studies and compare outcomes with different treatments after different treatments, then you lose some control of some effects. It's always easy to say if there's no difference, then it's like a more easy to conclude on. But if there is a difference, then you have to be really, really cautious on what has caused this difference. Is it truly a drug effect, or is it simply because there is a selection bias that you select different treatments to different patients to different treatments? And what I showed there was that the mortality was driven by causes that were unlikely to be related to rituximab maintenance, right? Or the lack of rituximab maintenance. For example, the fact that more patients that did not get rituximab maintenance died of colon cancer. Well, that would be a completely new indication for rituximab if this was to be true. And this is of course not true. This is a bias because you select patients that are more healthy for maintenance because you feel probably if you don't have another cancer, coexisting cancer, then there's more value to having rituximab maintenance. But if you have a more severe disease, serious disease, then your follicular lymphoma, then of course you go for some other treatment, or you change completely your priorities. But this example was really just to show that there are likely no survival benefit with rituximab maintenance. And even if you do retrospective observational studies and find the survival difference, then you have to be really cautious before concluding it, because there can be so much confounding. So really this was not to show that there is a difference. This was to show that is probably true. Like in the studies, there are no differences. It's just a survival. So it's just a selection bias that leads to a false difference in observational studies. This was my point.
Thank you.
Thank you, Chandran. We'll take one question from the audience. Dr. Arihant wants to know, in practice, while discussing treatment options with your patients, do you use real world data or the trial data?
I use what is available, and I think most guidelines are built on trial data as they should be. And this is, of course, the main decisions on treatments are based on trial experience. But as I also said, there's so many other aspects of being a physician that are not only the question on giving CHOP or R-CHOP or giving ABVD or BACOP. There's also like, how do you monitor patients after, for example? Do you need to do surveillance CT scan? Like explaining patients why you don't, because it doesn't add any value, but you give them radiation and things like this. Or do you need to do a biopsy, bone marrow biopsy, or there's so many aspects of being a physician that is not only about the exact selection of a specific chemotherapy regimen, where I think we rely most heavily on real world data. Also, for example, there's a question on using CNS prophylaxis in high grade lymphoma. Like, does it work? Then we can discuss with patients. There's some studies, real world data studies work, but you also have studies showing that it adds toxicity and we're not sure it works, and you prolong the time or you increase the risk of treatment delays in R-CHOP, which is really also maybe a problem, and things like this. So you can use this, I think, in in having an informed discussion with patients. And this is, I guess, this is what it's all about, that patients need to be involved in the decisions, because there, as I said, there are so many situations where there's no right or wrong, but there's pros and cons with different decisions, and in the end, only the patient can say what's right for them. So I try to discuss as much as possible, but of course, I know for you and for me as well, you don't have 2 hours every time you need to discuss decisions with a patient. You have limited time, so of course you cannot always do it in the detail that you actually wish to do, but at least you can try.
Thank you. Dr. Prashant. Unmute yourself.
Hello, sir. Thank you for this opportunity. The question I would like to ask is, you mentioned about the increased the bias against use of rituximab in some of the patients who might have comorbidities, and this might lead to the portrayal of increased overall survival in the real world. Counter to that, there are some studies, probably earlier this was reported in patients who got lenalidomide maintenance in multiple myeloma. There were reports of higher risk of secondary malignancies in those who got lenalidomide. And the explanation given at that time is that the secondary malignancies are actually popping up only because they are living longer with lenalidomide, and that's why you're not seeing it. So that was another kind of bias which was getting introduced. So how to account for, I was really interested in what you were talking about, this negative control outcomes. I want you to elaborate more on that in this context and how that would apply to something like this.
Yeah, that's a good example. I as I'm not a myeloma doctor, I cannot say I'm into the details of all the myeloma studies, but as I recall, there has been studies or meta-analysis of randomized trials that show a survival benefit of lenalidomide. Is that not true? Or
it is true.
Yeah.
It is true. Yeah.
Yeah. So this is a little different than from rituximab, because in rituximab the clinical studies did not show a survival benefit. For the negative control outcome, I think this is a very interesting idea that I came across almost by a coincidence some years ago. But I think this is really a nice way of checking your results if you can. This is more, it's not because what you find in the negative control outcome is a reportable finding in itself, but it's like a sanity check of, can this be true, right? And for example, this has been done in vaccines as well, vaccine studies as well. Like, you have so many suspicions on different side effects to vaccines, right? And sometimes you can do these negative control outcomes to really see if there's some kind of association that is completely unlikely. For example, this was used in a vaccine trial where they looked at the I think it was use of influenza vaccines, and reduction in hospitalization, and whether actually some of this difference, this effect was driven by the fact that there's simply different people choosing to get a vaccine, or not. Patients who get a vaccine against influenza take better care of themselves in general, and all these things. And this study, as I recall, showed also that the vaccines appeared to be preventing a hip fracture. And of course, this is a situation where you kind of, with all your medical knowledge, knows that this is unlikely to be true. And this is just a signal to show that probably this is not only a vaccine matter, this is also actually a matter of which type of patients that select which type of treatments. And that in itself is driving some of the outcome differences, and not the intervention, right? So I think this is how to look at it. Like a sanity check of what you find can really be, if that can really be true, right?
So, something like this, should it be built in when you are planning the analysis, or after you get the results, then you go back and recheck it by doing a negative control outcome?
I would say in situations where you have results that are clearly opposite of whatever has been shown before, then it's a good idea. And the other thing is that it, of course, also requires that you have the data sources to look for a negative control outcome, which is typically something that is completely different than the hematology outcomes, right? So we can do this in Denmark because we can merge all the registries. So I can also look at for example, in in patients with lymphoma, their cardiac history. I can also look at patients with lymphoma, which kind of, even that their tax payments or whatever, right? So because we can merge things like this, then we have the opportunity to look at really these negative control outcomes much better. But in other areas where healthcare data may be much more siloed and you have like different blocks and you cannot merge it, then it's difficult. Yeah, it gets more difficult.
Thank you. Thank you.
Thank you, Dr. Prashant. Dr. Tarak, I would like to have your remarks on one trial in follicular lymphoma upfront when the R-square was tried, lenalidomide with rituximab versus the usual treatment. They were looking at the superiority, and it came out to be that it is not superior, and it did not get the US FDA approval. Now, it is not superior, but it is not inferior. So should you use it in a clinical practice for patients who are not eligible for strong chemotherapy-based protocols, or you should not?
Well, that's a good question, and thanks for that. I would say, first of all, I of course cannot give recommendations against authority approvals. I would say for me, actually, we use very little lenalidomide upfront in follicular lymphoma, and we do that for the reason that we also often use, for example, RCVP for these patients. And in my experience, I don't find, at least not in elderly patients, I don't find lenalidomide that non-toxic. I think there's quite a lot of patients who get fatigue, tiredness, and quite severe myelosuppression, at least with the standard dose of lenalidomide in elderly patients. And these are typically also the patients that doesn't tolerate chemotherapy, right? But I think, in my experience, at least, I think treatments like RCVP, maybe they have inferior PFS compared to CHOP or bendamustine, but they are also, in my experience, very tolerable for elderly patients. So I haven't really felt a need to use this drug in first line, because I also see toxicities with that. What's your thoughts on this, Professor Agarwal? What's your?
So we also have not used it much. You're quite right. The advantage in India is that the lenalidomide is a very cheap drug, and it's a generic, and it's being a home therapy, then and rituximab just is possible on outdoor care. There are patient population who go for this. It has been in a certain way advantageous, and tolerance in the doses recommended for R-square, it's not bad, it's okay. So we have used it, yes, but not left and right, true. We go to the next question, Dr. Sonia Nityanan.
But I just want to comment, Professor Agarwal, because that's a really good observation also from the Indian perspective, because of course there are differences in healthcare that make different drugs like a better selection. And for example, we still have expensive lenalidomide, and in that situation, it's maybe selections are different. But also, to your point on the oral treatments, then maybe also now with COVID, this also of course have a huge advantage if you can stay out of hospital as much as possible at all. So that's a good point.
Thank you. Dr. Sonia Nityanan.
First of all, I would like to thank Dr. Agarwal for organizing this very important, lecture today. And Dr. Tarak, it was very informative for us, especially for clinicians. And I also would like to congratulate Dr. Akanksha Garg. So with this, my question to you is that from your talk, it's quite apparent that the real world data and the randomized clinical trials are complementary to each other. There is no place for replacement of randomized clinical trials at the moment. The two are complementary. Given that in a country like India, the real world evidence studies are easier to conduct and meaningful. Apart from being meaningful, it's much more easier to conduct the real world evidence studies than the randomized clinical trials. As you know, they're expensive and they need a good setup, etc. So it's a thought which I'm putting forward, and I would like to have your views for conducting these real world studies, to have them to be more acceptable with less, the main thing which is against real world data and real world evidence studies is the bias in them. So would it be more prudent to have a registration of real world evidence studies, to get them also registered, like the way one registers the randomized controlled trials, and have them done in a very transparent and open manner, so that it's very clear that when you change protocols or you deviate from protocols, that thing is very evident and very, kind of open and transparent. So what are your views on that? That was my first question, and then I'll take up a second question.
Yeah, I fully agree with you that this is important. And I think also there is a movement toward this being more of a demand. You can actually register your observational studies as well. This is the first thing. There's nothing against this. And then also there are also now like home pages where you submit your observational protocol. And this is also something we started to work on, where you actually publish it before you start the study, where you publish the protocol, where you publish the analysis plan, how you define your variables, because as I also showed in the beginning of the presentation, you make so many selections when you do a real world study, right? And maybe, or take so many decisions on how to define variables, how to use a cutoff, which data source, and all these things. And there are none of these decisions that are really wrong, but they become wrong if you change your decisions because you know the results at some point didn't work, and then you start playing with data and change your definitions and then suddenly, if you do this enough, then you come up with an interesting result. But this is of course then completely exploratory, and this has to be much more transparent, like what are you looking for and how you're defining things, and then prospectively. And if you don't find what you define, then you have a negative study as a rule, and then you can have some exploratory analysis, then you can confirm next time in your next study. This would be like in a randomized trial, and same approach in real world data would hugely, I think, increase the confidence in results generated in these studies.
Yeah, because in our country it's much easier to do these real world studies than to do large randomized control studies. That's why I brought it up. And my second question is actually a continuation of what Dr. Agarwal brought up, on his comments on lenalidomide. Like, if you are normally you do these real world studies or generate real world data after you have a published randomized control study data. But if one is looking, and that's quite okay when you're looking at the toxicity or side effects of the drug, you need real world data also. But going to the first part of your talk, which was focused on drug efficacy, is there a way to do a real world study, again, a real world evidence study for efficacy? And if something shows up on that, and then to document it with a randomized controlled trial? Because some things may be peculiar to certain ethnic populations in terms of not just the toxicity, but efficacy also.
Yeah, I think this is, again, a really, really good point, and would be really interesting. We would have some problems to do that just because of the fact that often it's difficult to get a drug before it's approved through a clinical trial. So we more or less always have the clinical trial, but you're asking for also like if you can.
So this is repurposing of drugs.
Yeah. Okay. Yes, of course you can do that for sure, and this could also be a very, very good way of generating new hypotheses if you have a tradition for using a drug in a specific situation that is a repurposing situation. I would say still a subsequent confirmation in a randomized trial would be a good thing then, because I think also, like the other way around, that we say that randomized trial cannot stand alone, then I would say also observed efficacy in a real world setting can also not stand alone for the selection biases that you always have, right? And you always need a comparator with a new drug, right? So if you have an experience in a real world setting with a repurposing of a drug, you need a comparator to show that this is really better than an alternative. And then you run into all the problems of, are you sure you can control all the confounders? Are you sure you have control of the selection and things like this? And in a perfect world where you have all information on patients, all data, everything collected, then of course you can do that. But the reality is that we typically do not have these informations, right? There's always some confounding that we cannot control because we don't know everything about the patients.
Thank you. Thank you again, Dr. Tarek.
Any other question from anyone? May I ask you one question? Yeah, please go ahead.
So my question is in continuation of what other people have asked. We do a lot of real world studies, but do you think there exists a bias in the journals in publication of these studies? Even if you look at one of the, New England Journal of Medicine, I don't think so ever, a real world study is published. Even for many other journals, they always prefer a publication of a randomized trial rather than real world studies. So do you think the international journals have to be a little bit more flexible in publication of real world studies and real world evidence? Thank you.
Thanks for the question. I agree with you that there is a bias towards randomized trials, but there is also a bias in general towards positive trials. And this goes, of course, both for randomized, but also for real world data studies. So in my experience, I think many journals, even the high impact journals, are much more open to real world data studies that are done in a very transparent way than they were maybe 10 years ago. And I think if you look at a journal like the Journal of Clinical Oncology and look at what's coming out of the Mayo Clinic in their observational cohorts, what's coming out and where this is published, for example, then I clearly see that high impact journals are also accepting real world data studies. Probably where there is a bias in some way is actually also on the source of the or the origin of the real world data study, because if you kind of have a real world data study, then it reflects a real world situation where you are, right? And this is not necessarily true the same way for a European country as it is for a US based real world study, right? So maybe this is actually also an important source of bias that the journals tend to take the real world data studies that reflects the real world that they are in. And if you have American journals, then, for example, maybe they are less likely to take European or Indian real world data studies because they will say, well, this is not the real world in the US anyway, so why should we read about it? But I think this is maybe a more serious bias on many journals that they tend to take studies out of their own countries more than on an international level.
Yeah, thank you.
Can I comment on this?
Yes,
Dr. Agarwal?
yes,
Yeah. So this is in continuation to what Dr. Pankaj Mehrotra just brought out. If you see, I think in New England Journal of Medicine 2021, there is this article which says, they say the magic of randomization versus the myth of real world experience. And sadly, it tilts towards the magic of randomization and not, the myth of real world experience. So it was like a little blow to us when we read this article, because I think the two are complementary to each other. And there is increasing awareness and data on the importance now of real world experience also. So I just wanted to bring that out. It is not a very fair article.
Okay. Dr. Tarek, I think, are there any more questions? Otherwise, I just put one last question. There appear to be none. So I just want your comments on various registration trials that are going on in last 1 decade or so, where the control arm is invariably an outdated, effective drug, but no more in practice today, and does not really form the standard of care in 2020 or 2021. And especially in the field of myeloma, this has been left and right. 2 drugs versus 3 drugs, 2 drugs versus 4 drugs, maintenance drug versus no maintenance, etc. Where if you look at the control arm, it is not in practice anymore because of its poor efficacy, and it was fine in that era in which it was developed, but no more. And the registrations are through, including of modern monoclonal antibody in myeloma and maintenance therapy getting changed accordingly. What are your comments on these kind of registration trials?
That is a really good question that of course I cannot really solve in depth because it's really complicated, and I see this from 2 sides. The first side is of course the one who conduct the trial. They are in a really difficult situation sometimes, especially I think in myeloma, but also in other diseases, because so many things are going on at the same time. And at the time you design your trial, and maybe it takes 2 years before you start, and then you finish after 7 years, then reality has changed, and you have no chance of really predicting how reality looks when your trial is finished and what kind of context your results will be put into. And I think when you look at the pharmaceutical industry perspective, I would say that they would have in my understanding, they would have no interest in selecting, for example, a very bad control arm. Of course, they may want to show efficacy and this is easier with a bad control arm, but they will have so many problems afterwards getting this drug reimbursed, at least in Europe. If you come with a study based on an inferior control arm that nobody wants to use, then all payers in Europe will say this is useless because we're not even doing this. We don't even know if it's better than what we do today. So I think this is the one side of it that it's. I think most will strive to give to have most best possible control arms. I hope they will, but I think it's so difficult to predict your future when you design the trial. And the other aspect is, of course, this is a situation where you may have a much stronger role for real world evidence, because you, at least when you get the trial results out and you feel that you have you're looking at a trial with a treatment that may be efficacious, but the control arm is actually not what you typically do, then at least some kind of comparison to what you actually do may be helping you in your decisions to see if it's really better than what you do today. Because you can say that the bias typically in real world data is that they are doing worse than clinical trials, right? So if you have an active arm with a new drug and you have your own experience in your own clinic or your own registries that show that what you do today is just as good, then the bias is probably towards that it's at least true, because typically, as I said, the trial will have better outcome just because of being a trial. So if it's not even better than what you do today or substantially better, then I would say that this could be a help to make decisions. But it's a very difficult topic because, as you know, there are so many drugs in development all the time, new things, change in strategies, change in treatment strategies. So 90% of trials coming out will look like they have some irrelevant control arm, but at the time they designed it, hopefully things made sense, right? So it's tricky.
One more comment I would like to have about use of brentuximab vedotin based upfront therapy for Hodgkin lymphoma. Your comments.
My comment is that we actually, I'd say in Denmark, we sometimes use it on an individual patient case basis for elderly patients where we feel they would not tolerate bleomycin. But of course, this is a situation, or would not tolerate bleomycin at all. But of course, this is a situation where a trial was ended up having a really outdated control arm in many ways when it was published, because the main driver of this treatment, because there was a minor PFS advantage but no OS, so the main driver would be to avoid bleomycin in patients, younger patients who can get severe lung toxicity on the long term as well. But today, like 80% of patients with Hodgkin lymphoma, younger patients, maybe a little more, get no bleomycin, no less than 2 cycles of ABVD, right? And this is maybe not a problem anymore. So why pay so much for a new drug when there is no toxicity problem, or at least it's substantially reduced now today with PET-driven decisions. So I think this is a good example of a situation where you may have an active drug, but reality just became better than the trial, much faster than they expected, the ones who designed the trial. So we don't use it for younger patients more at all in first line, and I would only use it in Denmark on an individual patient basis for elderly who I think should have something equivalent to ABVD, but I really don't dare to give them 2 cycles of bleomycin. So how is it in India? Can I ask?
Most of us cannot afford brentuximab upfront at all. So
yeah,
very little discussion on this side of the world.
Yeah, but I think this is no major loss in efficacy for Hodgkin patients, right? So,
I agree with you. Your remark was apt, absolutely apt. Any more remarks or questions for Dr. Tariq? If none, then I may just conclude by saying that it was phenomenal. Your intelligence was very obvious, both in lecture and in very thoughtful answering of the questions which the Indian faculty put to you. They were not easy questions, and you gave fantastic replies on the spot. Tremendous. I'm your fan now.
oh, thank you so much. Can I say a word?
All of us are fans.
Because yes. I just want to really thank you for inviting me. I really enjoyed it, and also enjoyed the questions. And I also want to say that we also, of course, have a lot of collaborations internationally on different studies, and these are typically also biased towards European, US, but I would love simply if some of you at some point just reach out if you have some ideas or want to collaborate, because I think really for good real world data studies, we need much broader representation of all regions of the world. Also in the same studies, we cannot do it only US European studies. This is only a minority of the world population. This doesn't apply to everyone. So please let me know, and because this would be both fun and really useful to work together on these things.
And I must say that the faculty that you have over here are virtually who is who. They're all in the position of conducting trials. They have numbers with them, and in general, India has numbers. And really, the real life data is on this, the real world data is on this side of the world. For the official vote of thanks, I invite Mr. Ashish.
Thank you, sir. We at Zydus sincerely thanks MHG and Dr. M.B. Agrawal for giving us this opportunity for supporting a webinar on such a relevant area. I thank Professor Tariq for his great session today, and through his session highlighting the relevance, need, and importance of real-world evidence. Through his lecture, it's clear real-world data has its own significance and can help clinicians with various relevant data sets which can improve clinical outcomes. He also highlighted why outcomes from real-world data in clinical trials can be different, and I thank him sincerely for his session today. Thanks to all the eminent faculty for a wonderful discussion, and not to forget our respected virtual delegates who joined us today afternoon. I once again thank Professor Dr. M.B. Agrawal for his untiring.
I think we lost Mr. Ashish. Well, thank you very much to everybody, and have a wonderful day ahead.
Thank you very much. Thank you, sir.
Thank you, sir. Bye.
Thank you.
Thank you,
Bye.
Dr. Tariq, again, and thank
Thanks
you,
for
Dr.
inviting.
Agrawal.
I really enjoyed it. Thanks a lot.
Thank
Thank
you.
you so much.
Bye.